How does epigenetics influence brain development? Have you ever wondered how our genes and environment shape our brains? Today, we will discuss how epigenetics plays a role in brain development, especially in the context of schizophrenia. Epigenetics involves chemical changes that affect gene activity without altering the DNA sequence itself. These changes include DNA methylation, histone modification, and regulation by non-coding RNA. During brain development, these epigenetic changes are essential for controlling when and where genes are activated or silenced. This regulation is crucial for forming neural cells, differentiating them, and establishing connections between them. Proper brain structure and function depend on these tightly regulated processes. Now, let's connect this to schizophrenia, a complex mental health condition that has a strong genetic component. Schizophrenia does not arise from a single gene but rather from multiple genes that each have small effects. Epigenetic modifications can influence how these genes behave during critical periods of brain maturation. One key mechanism is DNA methylation. This process adds methyl groups to DNA, which can suppress gene expression. In individuals with schizophrenia, researchers have found altered DNA methylation patterns in genes related to neurotransmission and brain development. Such changes can disrupt normal neural pathways and cognitive functions. Another important mechanism is histone modification. Histones are proteins that help package DNA. When these proteins undergo chemical changes, they can either promote or inhibit gene expression. Mutations in the enzymes responsible for these modifications have been linked to schizophrenia, affecting genes that are vital for synaptic transmission. Non-coding RNA also plays a role in regulating gene expression after transcription. When these non-coding RNAs are dysregulated, it can lead to abnormal brain development and has been implicated in schizophrenia. Environmental factors, such as prenatal stress, infections, or low oxygen levels, can trigger epigenetic changes in the placenta and fetal brain. These changes may increase the risk of developing schizophrenia later in life. They can affect brain regions that are crucial for cognition and emotional regulation. Understanding how epigenetics influences brain development can lead to better early diagnosis and targeted treatments for schizophrenia. Epigenetic markers could potentially serve as indicators for identifying individuals at risk. Additionally, therapies aimed at modifying epigenetic states might help correct abnormal gene expression patterns associated with this disorder. In summary, epigenetics plays a vital role in shaping brain development by regulating gene expression in response to both genetic and environmental factors. In the case of schizophrenia, these modifications contribute to the onset and progression of the disorder, highlighting the complex interplay between genes and the environment.